today is Exome is great, but Genome is better. So through our talk, we are trying to show how exome sequencing is useful in clinical practice and how whole genome sequencing has allowed to us find a disease-causing variant that more routine tests, including exome sequencing, could not find. So we'll have 3 talks lined up. So our first two talks will be presented by our longtime collaborator, Dr. Mangli from Sheffield University and Dr. Kamalaji from Tbilisi Medical University, who thankfully agreed to join us today. So they will share the couple of cases diagnosed by Exome and how they use this information for clinical management of their patient. And then I'll present on the clinical utility of the whole genome sequencing. And before we get into session though, I'd like to give the brief introduction of our company. So as I said before, our company name is 3 billion. So I think this audience will appreciate the meaning of it than my non-genetic friend who interestingly think about money when they hear the 3 billion. So actually 3 billion is the number of the base pair in entire human genome. So as a genetic diagnostic company, for rare genetic disease. So our primary goal is to find correctly and quickly find the disease-causing variant out of 3 billion base pairs. So our company was founded in 2016 by our current CEO, Chang Eun-gun, with the vision of providing affordable service for patients with rare genetic disease. Our mission is to ensure that no undiagnosed patient is left behind without access to genetic testing. So one fun fact is about our CEO is that his birthday is Korean Rare Genetic Disease Day. So every year on his birthday, he believes he's destined to work for rare genetic disease. So okay, what do we offer? We more accurately genomic testing for patients with a suspected rare genetic disease. This table summarizes our test menu. So we offer genome and exome and variant-specific testing for genotyping family members of exome and genome patient. So 3billion is done by Sanger sequencing. So we have accredited by CAP, College of American Pathologists in 2021. We also received in CLIA in 2022. So all of our tests run following the CLIA and CAP standard. So this is 3 billion's workflow. So entire workflow from specimen to reporting has been validated as one whole test. Okay, sorry. So one sample is arrived at, we generate the report no more than 6 weeks. So this is 3 billion's pipeline. So in my talk later, I will delve into more detail. So I'd like to mention about the most important and cutting-edge technology we developed, evidence, and 3S and automated daily reanalysis system. So first, evidence is a variant prioritization system which is composed of 3 key modules. It's a variant annotation module with daily updated database, customized variant classification module, and phenotype similarity scoring module. So we follow the ACMG guideline for variant classification and we further customize it to make it more robust. And we are updating our database as soon as there is a new release to make sure that we are using most up-to-date information for variant interpretation. And since we are searching the variant for rare genetic disease, common variant get filtered out except no pathogenic variant, and remaining variant get sorted by symptom similarity. And second, to support and accelerate manual review process, we developed a variant recommendation system, which is called 3Ask. This system mimics the steps medical genetics would go over when reviewing each variant. So 3Ask is data-driven machine learning model that incorporate ACMG Bayesian score, variant quality score, and similarity score to suggest the variant that are more likely to explain the patient phenotype. So as you can see the left graph, the 3Ask represented by red bar outperform other variant recommendation tools such as LeaderCon and Exomizer that you may have heard of. And so as you can see the right graph, when retrospectively tested on 3 billion data set, 95.7% of the variant are within top 5 recommended by 3Ask. So before we talk about the real analysis system, let me share our overall diagnosis rate. The overall diagnosis rate is approximately 30%, but as you can see this graph, the diagnosis rate can vary quite a bit depending on the disease category. But even for the diagnosis rate with the highest yield and despite the best effort, more than half of the patients still remain undiagnosed. So why is this? Well, there are several reasons for that. So first, it is possible that certain patients don't have genetic disease. And secondly, it's also possible that certain variants are not identifiable by current NGS technology or not interpretable by current bioinformatics tools. And lastly, causal genes may not have been discovered yet as a disease genes. So that if the last case, though, reanalysis of existing data can be necessary to find the disease causing variant once causal genes have been discovered. So that's why the reanalysis can be critical to find the disease-causing variant in undiagnosed patient. So how often should we do the reanalysis? Most labs run routine reanalysis on exome 1 or 2 years after initial analysis done. So we thought that why wait so long if you can implement more frequent reanalysis in an efficient way? So 3billion has implemented automated daily reanalysis system since 2019. So we run evidence every night annotating all variants from undiagnosed patient using update our database and classifying the variant accordingly. And our medical genetics team reviewed all variants that were reclassified as pathogenic or likely pathogenic in genes that could fit the patient phenotype. So this is actual case. Actually, the 2-year-old boy showed neurodevelopmental delay and dysmorphism, but the exome initial report was negative in February 2021. So 1 month later, one paper was published. In that paper, multiple variant in SPEN gene was reported in 13 individuals with developmental delay, dysmorphism, and multiple anomaly. So and then our patient also had nonsense variant in these genes. So I compared the patient phenotype and determined that it could fit well. So we generate the report as inconclusive because this evidence is insufficient to define gene and disease association. So but 2 months later, in May 2021, this gene was registered in OMIM being associated with radio-Tartaglia syndrome. So finally, we are able to generate the possible report diagnosing patient with radio-Tartaglia syndrome. Okay, so before I introduce our first speaker, let me close by briefly going over some of our major achievements. This is our sample growth rate. So we've been tested more than 40,000 samples in the past 3 years. So we also aim to double this number each year. But the more patient data we accumulated, the more accurately that we diagnose each patient. So, and I'm very proud to say these patient data come from over 350 medical institutions across 50 countries. And also, we are contributing to the genetics community by participating in the national projects, such as development of AI-based software for variant interpretation and establishing a national genomic database using WCS, and also publishing paper with collaborator and a physician and filling the patent and submit the variant to the CleanBar, one of the public database. Okay, that concludes my introduction. So as I come back later for my last talk, so I'll take a question after that. It's okay, so without further delay, so I'd like to introduce our first speaker, Dr. Manglio. Dr. Alistair Manglio is a senior clinical lecturer in neurogenetics at the University of Sheffield and honorary consultant clinical geneticist at Sheffield Children's Hospital NHS Foundation Trust. He is a clinical lead in neurogenetics, so he will present a topic on genotype and phenotype correlation. Dr. Manglio, so it is a great pleasure to have you speak. Thank you.
Very unusual to have 30 degrees heat in Glasgow at this time of year, or at all. these are my declarations of interest. Perhaps notably, I'm the editor-in-chief of the European Journal of Human Genetics, which is the official journal of our society. So exome sequencing is based around exons. So what is an exon? Sorry. I asked ChatGPT what an exon is. If you ever use ChatGPT, you'll find out it gives you a massive long answer of variable quality until it hits upon a reasonable answer. And this is sort of a reasonable answer for what an exon is. So it's the bits of the genome that encode a protein. But also, it misses the trap we all fall into, because towards the second last paragraph, it does point out that there are regulatory elements within the exons. And the experts in all this, Nicola Whiffen and Julie Aspidin, published this year a viewpoint in Cell Genomics, correcting us all. So whole exome sequencing does not capture every element of the exon. It misses out the 5' and 3' UTRs. And we should think of clinical exome sequencing as sequencing the CDS. I've forgotten what they call CDS, coding sequence. So even in the best exon sequencing, we don't really capture 5' and 3' UTR variants. So what's a gene? As we said, a gene is something that encodes a protein, consisting of introns and exons. And even in the best healthcare systems, what we're really looking at is the exonic elements and perhaps some canonical splice sites. Now in the NHS, we've got whole genome sequencing. And if you wait long enough, you'll get a report from Expansion Hunter or a similar algorithm looking at the trinucleotide repeats in the intronic regions. But the limitations we have is that we don't really understand how to report 5' and 3' UTR variants in clinical practice. In our clinical labs, we're not scientists, we don't have functional work set up. We have to base ourselves on what's in the literature and what we can do informatically. So why did I tell you that? Everyone thinks genome sequencing is a lot better. Technically that's true, but in terms of the effort you put in my opinion, most of what we can interpret comes from an old-fashioned clinical exome, and you could spend 5 hours trying to interpret genome data and get one more diagnosis, or you could work your way through more exome sequencing. My favored solution is to use more exome sequencing, but that might not be right. So many of you will know this. There's 3 broad types of test. The gene panel test, which reads at reasonably great depth a small number of genes, which is great for a defined phenotype like breast cancer or hypertrophic cardiomyopathy. And at the other end of the spectrum, we've got whole genome sequencing, which is said to cover greater than 95% of the genome at moderate depth, captures all the exons, introns, promoters, and UTRs, but then the challenge is interpreting all that. And in the middle, there's exome sequencing, which covers 1 to 2% of the genome, depending on how modern the exome is, at a greater depth of coverage. So exomes are possibly more likely to pick up mosaic variants because they've got greater depth. Genomes can still pick up mosaic sequence changes in blood, but less accurately. So if you want to look for a mosaic change, you really have to use a bespoke panel with ultra-deep sequencing. So that's a consideration if you've got a negative exome or genome. The trick in the NHS is that we do whole genome sequencing, but we only report a panel of genes for the relevant phenotype. So politically, it's great to say we have a genome medicine service. What we've really got is a gene panel service. And that can catch people out, especially mainstream clinicians. So an illustration of the gene panel problem. This is a 16-year-old man who came to clinic in genetics because he transitioned from pediatric to adult neurology. He essentially had a hereditary spastic paraparesis with dominant inheritance. And the neurologist did the correct thing. They did the genome sequencing panels for pediatric and adult onset spastic paraparesis, which was negative. And then we were lucky enough to be able to offer him a research exome through 3billion, and he's got a heterozygous known pathogenic variant in BIC-D2. BIC-D2 is an unusual condition because it's a distal spinal muscular atrophy. But if you look at the original papers, there are 1 or 2 families where the presentation was with a spastic paraparesis. So the patients don't always read the textbooks. Sometimes they present with atypical phenotypes. And I think in this family, the BIC-D2 variant was explaining his spastic paraparesis. And if we hadn't done an agnostic exome, we'd have missed that, because BICD2 in the NHS is not taken to have enough evidence of an association with HSP to report on an HSP gene panel. We've got relatively restrictive criteria. You have to have at least 3 separate families with a disease-gene relationship for it to be reported on an NHS gene panel. It's impossible to do that accurately for every single gene. We can't search literature for everything. And in the NHS, Panel app, which defines what diseases go on what panel, was crowdsourced, done through an online application system where people voted as to whether a gene should be on a panel or not. And there's inherent bias sometimes in what's on the panel. And some voices shouted louder than others. So if you're running an NHS test, be very aware of what's on the panel, and perhaps don't take no for an answer if the lab tells you there's no diagnosis. An even more frustrating issue is intellectual disability. This was a lady in her 30s who had intellectual disability, but she was independent enough to sign her own forms for genome testing, and she had a number of problems, including sensorineural deafness and hypocalcemia. So I first thought she had a 22q deletion, but C and V's were not present. So we've got this massive R29 intellectual disability panel, which can be read from genome sequencing in the NHS, and that was negative. And there are so many genes on it that it's impossible to work out what is and what is not on it, unless you're looking for specific genes. So again, we were lucky enough to be able to run a research exome for this lady, and she's got a GATA3 pathogenic variant, HDR syndrome, hypocalcemia, deafness, and intellectual disability. And for some reason, initially this was on the hypocalcemia panel, but not on the intellectual disability panel. It's been corrected now. I can't work out the logic of having this on a hypocalcemia panel, because these patients don't come in with hypocalcemia, they come in with dysmorphism and learning problems. She also had malformations of her uterine tract, which are a feature of HDR, and she had renal cysts. So this made a management difference for this lady. She had follow-up which detected structural abnormalities. So you've got tight genotype-phenotype correlations, that's great. You can be confident in your gene panel. If it's something like epilepsy or intellectual disability, which has got such allelic heterogeneity, I think you have to be very cautious using panels. And of course, you have to be aware of what platforms can pick up CNVs and what can't. Ideally, you'd run a SNP array before doing exome sequencing if you can. There's no reason why exome and genome panels, pipelines can't pick up CNVs. It depends on the informatics. Okay. So how do we get around this tremendous phenotypic heterogeneity and allelic heterogeneity in genetics? Peter Robinson and the Monarch Initiative almost 15 years ago started to solve this problem by coming up with human phenotype ontology, HPO terms, and I think there are now over 15,000 HPO terms. So it's very comprehensive, but actually for a non-expert somewhat difficult to use. It's an ontology, so it starts at the top with an organ system, then a part of the organ, then a specific malformation. So we now can, in a computational way, describe a patient's phenotype in a great deal of detail and granularity. And that helps us interpret and annotate genomes and exomes. So because of HPO terms, we can use genomes and exomes in an agnostic way, saying we don't know what the diagnosis is, but we think there is one. So typically with an exome, we can cover all 20,000 genes, greater than 100 times coverage, and now 3 billion also cover the mitochondrial genome. And the concept is that we use phenotype-informed diagnostics with the evidence and other pipelines that my colleague was mentioning. So the principle is you first get genomic data, identify pathogenic variants, so ACMG class 4 or 5, through standard features such as low prevalence in the general population, CAD score, Splice AI, whatever your favorite tools are, and you rank them 1 to 10, 1 being the gene with the most pathogenicity, 2 the second most pathogenic evidence. And then you use the HPO terms, and the computer programs ask, for these genes, have they been linked to any of the relevant HPO terms? And there are a variety of bioinformatics pathways that were used in the original Exomizer software, including human descriptions, mouse models, zebrafish, fly models. And these pipelines are surprisingly successful. Exomizer is what we use in the NHS. It's probably the best known. If you analyse only the top hit, you'll get a correct diagnosis in about 70% of cases. And if you go down to 10, the top 10 ranked genes, you get a diagnostic rate of 80%. If you go any lower down the scale, it just tends to be a lot of noise and difficult to interpret. So this is how you manage a large volume of variants coming from an exome or genome. Use something like Exomizer rather than restricting yourself to a gene panel. And the performance of the evidence pipeline from 3 billion is very similar. After you've done trio testing and phenotypic analysis, almost 90% of patients who went through got either a pathogenic or likely pathogenic variant identified, which is great. Okay. Some other examples. Let's finish some clinical examples. This was an adult female who presented with short stature. She was 5'3". We were asked, does she have Turner syndrome? She had 2 children, so the answer was no. She was dysmorphic. She had sensory neural deafness and very severe aortic coarctation, which was being worked up for surgery. It wasn't really clear a unifying diagnosis, even after an MDT. She was in one of our peripheral hospitals, so accessing imaging and skeletal surveys wasn't appropriate. So an agnostic whole exome sequence identified biallelic variants in ARSB, which is the gene for Martu-Lamy syndrome, type 6 MPS. So this is usually a very severe pediatric onset condition, which can be fatal. But there is an expanded phenotype of very mild adult onset forms, and we confirmed this biochemically, and she's been referred now for enzyme replacement therapy in the National Lysosomal Storage Disorders Clinic. So hopefully that will stabilize her aortic coarctation, the aortic stenosis. But to get to this point, without a diagnostic test, it would have been very difficult. It would have taken several months to wait for the skeletal surveys and biochemical testing. And it probably cut 6 months off of our time to get into treatment. Another sort of similar story, a non-specific presentation in a SIB pair of adulthood with very severe refractory inflammatory bowel disease and thrombosis. And the histology was most unusual, I was told. It didn't look like Crohn's or ulcerative colitis, and it hadn't responded to the standard effective immunomodulators for this. So we did whole exome sequencing, and they found biallelic deletions, frameshift deletions in a gene called CD55, which is something I'd never heard of. It's Chapel syndrome, which is a recessive cause of inflammatory bowel disease due to CD55 deficiency. And if you look at this initial paper, it's fascinating. They have an upregulation of certain components of the complement pathway, and monoclonal antibodies against CD55 somewhat normalize the peripheral blood inflammatory markers, so it's potentially treatable. So this lady and her brother have been worked up for treatment with a monoclonal antibody. It's off-label, so it's not entirely clear whether they'll be allowed to have it in the NHS. But if she'd had this at disease onset, then it would have prevented years of morbidity, surgery, and actually potential death from the thrombosis she's suffered. So I think if we do pick adult patients from highly selected cohorts in teaching hospitals, there are genetic diagnoses to be made, and they do alter treatment. And actually, their presentations can be not terribly exciting. They can look very bland, because a lot of the dysmorphology and characteristic clinical features in pediatric cases might be obscured in adulthood. And a gene panel might not pick this up because we didn't have a good phenotypic handle. And CD55 is not on the immunodeficiency panel, which you can access in the NHS. Okay. Another one, oh, so this is wrong. An adult patient with diplegic cerebral palsy was referred to us, and I'm sure you've all seen this now. I thought cerebral palsy is actually genetic, and this gentleman had a spastin pathogenic variant, heterozygous spastin pathogenic variant, which is the main cause of spastic paraparesis in Western populations. But there's no cerebral palsy gene panels on the NHS. It's not a recognised presentation of a genetic condition. So our lab would probably have rejected that referral, which is why we went down the research exome route. Okay. So what's our message? Doing an exome or a genome obviously increases your diagnostic rate compared to a gene panel alone. You have to be aware of how your exome or genome is being interpreted. If someone's just applying a panel test to an exome or a genome, it's just a panel. If someone's using Exome or another phenotype-informed pathway, then it is a true agnostic test. If you've got pediatric cases, I think trio is the best approach. It does increase the diagnostic rate. If you've got an adult, the chance of getting a trio approach is less, but it doesn't seem to make a difference in adult patients. We still seem to be able to diagnose them quite confidently, mainly because it's not intellectual disability. Be mindful of what's not on panels, genomes and exomes. The classic catch is where adult neurologists are referring their patient for testing for ataxia or Parkinsonism, when actually they've also got intellectual disability. And many adult neurologists, unfortunately, don't recognise that most intellectual disability syndromes can also cause a movement disorder, and diagnoses are being missed because you need to include the intellectual disability as well as the movement disorder on your request, at least in the NHS. So thanks for letting me speak. I will acknowledge people at Sheffield who helped me with my work, particularly 3 billion, who've given us access to research exomes in a time of restricted funding in the UK. And Peter Robinson, who's had some very helpful discussions with me very kindly about HPO terms. Thank you. Should we take questions now, or do you want me to wait till the end? No questions. Any questions?
Okay, thank you for interesting talk. Okay, our second speaker is Dr. Tinatin Kamalaji. So Dr. Tinatin Kamalaji is from Tbilisi State Medical University, Georgia. She is the head of the Department of Molecular Genetics at Tbilisi State Medical University and a clinical consultant in several major pediatric hospitals. She's a clinical lead in inborn areas of metabolic disease. So she will present it on doctors are great, but exomes are better versus exomes are great, but doctors are better. Thank you so much, Dr. Tinatin, for joining us.
So good morning, and thank you for inviting me as a speaker today. It's my honor and pleasure. So I chose to go with intriguing names, which are better, exomes or doctors, because we live in the era of fascinating technologies, but I still think human brain is even more fascinating. So we live in the era of new gene discovery. In PubMed every year, the new gene discoveries publications are increasing with incredible speeds. For Mendelian disease, and I really like this diagram of updated epilepsy genes. And of course, this is driven by technologies, and today is the era of precision medicine. Without diagnosis, we cannot treat, and treatment, I think, is possible as we understand the molecular basis and pathophysiology, all of that. So, exomes and doctors. Exomes have untargeted approach to look for disease-causing genes, whereas doctors have targeted approach to look for specific diseases. Exome looks for the known genes, and doctors look for the known diseases. Exomes allow gene discovery, whereas doctors allow evaluation of the new phenotypes. And exomes detect new genotypes of the known diseases, and doctors detect new phenotypes of the known diseases. And with exomes, interpretation is not possible without clinical data. And for doctors, diagnosis is sometimes not possible without exome because some diseases are very heterogeneous. And of course, there is no one perfect genetic test to see all rare diseases causing DNA alterations. Maybe in a couple of years we'll come to that, but not now. And there is no perfect doctor that knows all rare diseases. So sometimes other specialty doctors than geneticists may say, okay, just do exome and you'll find the gene. Okay, but clinical drives finding interpretation. And I have like some examples of when doctors are great, but exomes is better, and then reverse. And then we'll come up to the conclusion of how it is. So an example of the consanguineous family with affected sister and brother, and the children have developmental delay, intellectual disability. You see skin manifestations of psoriasiform dermatitis and plantar keratosis, dry skin, alopecia, polydactyly in the sister. And somehow we had the differential diagnosis with ectodermal dysplasia, but going through the differential diagnosis, actually none of them fitted with polydactyly or intellectual disability or the type of inheritance with any known ectodermal dysplasia syndromes. So Exome was performed, and you might have not heard about this gene because only 6 patients have been described so far in the literature with MSM01 gene in the homozygous state, and it's a methylsterol monooxidase 1 deficiency. It's a gene that's involved in distal cholesterol metabolism. And the treatment involves cholesterol supplementation, statins, and bile acids. And if you compare, we started modified diet treatment for these patients, and the skin improved, the hair improved, and just in June, it came out our publication in Clinical Dysmorphology Journal. So this is when the exome gives a diagnosis, and we understand that some metabolic conditions are treatable, so we should never miss a treatable condition. Another case with a child, with brothers, 12 and 9-year-old brothers, who have delayed motor development, deficient gross motor development, actually normal intelligence, they go to school, and non-dysmorphic, but the major feature is non-progressive ataxia, which worsens during illness. These are 2 strabismus. So somehow Friedreich ataxia was in the differential diagnosis, but since the children do not have progressive ataxia, Friedreich was not a very good differential diagnosis. And then many triplet repeat ataxias are dominant, so because we have brothers, we were more prone to think that it's something recessive. So went for the exome, and interestingly, PMM to CDG, 2 pathogenic variants. And I really wondered because there were no fat pads, no dysmorphia, no liver problems, no intellectual disability. I even doubted whether this could have been really a PMM to CDG. So what to do next? Do reverse phenotyping, which is very important. So brain MRI showed diffuse cerebellar atrophy, and carbohydrate deficient transferrin was abnormal, which proves the diagnosis, plus with S protein and antitrobin. So they're on the follow-up with the neurologist, and they have improvement in their ataxia with acetazolamide. And of course, the family received the genetic counseling. And another case, a 6-year-old girl from consanguinity, global developmental delay, intellectual disability, regress in development, movement disorder, and unexplained elevated ferritin. Exome was done, and PLA2G6, homozygous pathogenic, which is causative for Parkinson's disease, and this explains the regress in development, and then HFE, which is causative for hemochromatosis. So the girl has dual diagnosis. And interestingly, father, I highlighted over here, father is also homozygous for hemochromatosis. The mother is heterozygous and the father is homozygous. So that's why sometimes parental testing is really useful. And father has never done ferritin analysis before. So we again went to reverse phenotyping, and he's under the supervision of the doctors. So this family received genetic counseling in regards to 2 conditions. One is milder, of course, with hemochromatosis, but with Parkinson is much more severe. So kind of take-home messages is that exome is the first-line investigation in unexplained NDDs. Some NDDs are treatable, so we should never miss a treatable condition. Exome expands the phenotypic spectrum of the known diseases. Sometimes we may not suspect because mild spectrum of the disease may be very unspecific. And negative exome is not always negative. And there are new genes that are continuously discovered. So we should not give up and still continue pushing these patients for the diagnosis. And again, dual diagnosis is something that is not uncommon. Now, exome is great, but doctors are better. And since I'm the clinician, in our country, we don't have access, on-site to do exomes, we really try to have a strong clinical evaluation to drive good interpretation of the exomes. So, 5-year-old boy with delay, intellectual disability, lens dislocation, thrombosis, elevated homocysteine. So, not a difficult diagnosis to guess and to have a strong suspicion of the homocystinuria. And exome was negative. What to do next? Right to the lab, because if you have a strong clinical diagnosis and strong suspicion, this could not have been anything else than homocystinuria. And I wrote to the lab, got a reply that we have found evidence of a small deletion across exon 3 of the CBS gene, and then we understand that sometimes exonic deletions are something difficult to pick up with certain workflows of the exomes. And we will send out a new report once the confirmation is complete. So we got an updated report, and whatever was suspected, that was proven. So negative exome is something that if you have a strong clinical suspicion, go ahead and push the lab. Another boy, 4-year-old with intrauterine growth restriction, delays, kidney hypoplasia, brachial fistula, periauricular tag, and cryptorchidism. And Bohr syndrome, actually mother is a doctor, and she came up with, and she said, okay, my child has Bohr syndrome, and I want genetic test to confirm that it is really that. And we know there are 3 genes causative for Bohr, so exome looks a good option for those conditions that are genetically heterogeneous. You see the results? Of course, Bohr's is confirmed, but plus the lab reported 2 PAH pathogenic variants. And could this have been a dual diagnosis of Bohr's and PKU? And then newborn screening was normal. The mother did not receive any notification after the newborn screening. What to do next? Reverse phenotyping, of course. And of course, measurement of fat levels is something must-thing to be done. And there's a really nice database of PKU mutations. So you go there, type the variant, and then it gives you different features, including enzymatic activity, which is very important when we talk about PKU because it's an enzyme, and enzymes have different activities. So this is variant 1 and variant 2, and we see that one variant has 28% activity, another 65%. So total, that would count up as 46.5%, which is similar to heterozygous unaffected parent of the child, for example, who has PKU. And levels were also within normal range. So even though the lab reported that it's PKU, it's pathogenic, doing good reverse phenotyping and making genotype-phenotype correlation excluded, in this case, the diagnosis of PKU. Family. This is really one of my favorite cases. You see a family of the father, of the daughter, of the son, and you see the skin manifestations, the brain MRI, angiofibroma, so a very clear-cut tuberous sclerosis diagnosis, right? Even it's a familial one. And another way why I'm really fond of this family is that it reveals the clinical heterogeneity of TST, where father has normal intelligence and the daughter has severe epilepsy, and the son does not have epilepsy, but he has some delay, so it's a very heterogeneous disease. Exome was negative, and of course, no one would persuade me this was not TST. But the exome reported of the father having BRCA1 pathogenic variant. What to do next? push the lab, of course. And I remember I had to write like 3 emails to the lab for them to do MLPA, and actually they detected the deletion of exon 1, and exon 1 is a non-coding exon. So by definition, that would not be excluded in the exome workflow because it's a non-coding exon, and it's pathogenic, and the father is 21% mosaic. So this mutation happened in him after conception, and plus he has BRCA1. And the daughter and the son both inherited TST2, but not BRCA1. So the father is kind of a double times prone to develop certain cancers, whereas children are not. And basically, for the next children, it's having 2 pathogenic genes in the whole family. And actually, cardiology and oncology genes are not uncommon incidental findings in the exomes, and they're medically actionable. So, some take-home messages. Negative exome, of course, is not always negative, and we should think broader. And for me, is in everyday practice, when I get positive result, that's like the easiest because I have the diagnosis, I counsel. VUSs is a challenge. I did not give any examples of VUSs because the format and the message was a little bit different for today. But when it's negative, it's something when you think, is it really negative? Or is it not negative and we need to go for other option, or write the lab or choose another type of methodology to have the diagnosis. And some West workflow have limitations in detection of exome level CNVs and triplets and not coding regions. And of course, interpretation of exome results should always be in the context of the clinical findings and with reverse phenotyping. And good clinical information is crucial for interpretation. And doctors, be bold, push the labs to find the mutation if you are sure of your clinical diagnosis. Because the people who sit in the lab, they don't see your patient. You see your patient, so you need to be defender of your diagnosis and of your patients. And of course, exome is useful for detection of the secondary findings for the NCMG medically actionable genes. So these are the exomes, the Lego bricks, and doctors give the instructions. And then we assemble the big piece of Lego. But we need to find the missing brick, a mutation. And this is only possible if we get together. And these are the hands of my children and my mom. We took it at home. And these are my fantastic colleagues from my university. Thank you. Have any questions? I'm happy to answer. Yes, please.
Just a comment on your second category. Exomes are good, but doctors are better.
Yep.
So the example that you gave in phenylketonuria, and one variant had 68%, and the other variant had 28%. The database should have classified the 68 variant as a benign variant. So that should not have been reported anyway. So you should not have gone to that path to calculate yourself. So was that a deficiency of the reporting lab? That actually is not telling you that exomes are not better. Exomes are still good. The other example.
May I answer you for this in regards to PKU? Because sometimes someone may have this 62% activity and another null allele, and this can result in variant PKU. So I think that's why they still classify and report the high activity allele as pathogenic, because maybe in combination with another null mutation, it could still give the phenotype. But our combination was 26 and then 62, so as a summary, but now I always check with PK. Whenever I have PKU and PAH, I always go to the database to check for activity.
But exome is not just machine.
Of course.
Is a result given by an accredited molecular pathologist. Is it their job to calculate this, verify the database, and then issue a report? Instead of the clinician calculating, is it doubling up the effort?
Yeah, it depends from lab to lab. I've had reports from the other labs where they state the enzymatic activity, and that was really nice. But then this lab did not report of the enzymatic activity. So it depends on, I mean, you as a clinician do not only need to rely on what the lab writes, but you should judge and give your own interpretation. Of course, good reports are always very helpful. I agree.
From my understanding, can you tell me the mechanism of non-coding deletion? How was it pathogenic?
In TST1, right? Because it involves the deletion of exon 1 and plus some part of the promoter. So the transcription would not be initiated if you have deletion of very important, so to say, starting point of the gene. So it's pathogenic. And the report also stated that in their internal database, they had a similar upstream non-coding deletion in 1 or 2 other patients. So it's not published, but in the lab's internal database. So because of transcription initiation, that's pathogenic, yeah. Thank you. Yep.
Thank you for your inspiring talk. And do you have any strategy for diagnostic method? Just after the exome sequencing is negative, do you follow the genetic analysis periodically or something else?
Yes, yes, of course. So there are some negative reports where you have some perinatal complications, so you can blame on that. But then if your patient is like too dysmorphic or if there are too many things and exome is negative, of course, it's always easier for me as a clinician if I have a strong clinical suspicion of certain group of syndromes or a certain syndrome, then it's much easier to push the lab. But sometimes your patient is not like that. So I've had a case with a child with microcephaly, very severe delay, and 2019, 2020 exome was negative, and then I sent the blood at 3 billion, and then we received the gene in 2022, and the gene was discovered in 2021. So it's very important to follow up your patients and then either do reanalysis or do new exome or genome, because new genes are continuously discovered. And if you have a negative report at 1 year, following up those patients and pushing them for further investigations can give you diagnosis, not because the lab was bad, but because the gene was not yet discovered. Yeah. Okay, thank you.
Okay, thank you. Okay, so let me introduce myself to you. So those who came in later, my name is Gonsa. I am Chief Medical Officer at 3 billion. So actually, before joining this team, I worked as a pediatrician and clinical geneticist 4 years at Asan Medical Center, which is one of the big hospitals in South Korea. And then I believe that my background in pediatrics and clinical genetics has given me a unique perspective on challenges and opportunity in the field of genetic testing. Okay, enough about me. So this is the title of my talk. So I'll talk about the clinical utility of whole genome sequencing using 4 cases diagnosed by whole genome sequencing. So as the Dr. Mangli described earlier, so let me briefly go through the differences between the panel, exome, and genome sequencing. With the panel, we can sequence a subset of the gene or a specific disease or collection of the disease. So it is very usable when the suspected disease is very specific, but it is possible that certain genes are not included in the panel, especially recently discovered gene. The whole exome sequencing targeted protein coding exon lesion of almost genes. So since the majority of known pathogenic variant within almost coding exonic lesion, so it is a very cost-effective way to find the disease-causing variant when many genes need to be tested simultaneously or when it is difficult to specify genes that need to be tested. And whole genome sequencing, as you can see this figure. So whole genome sequencing entire genome. So the coverage is very uniform across the intron or intergenic region. So this allows us to uncover the variant in intronic or intergenic region that potentially significantly alter splicing pattern or expression pattern. And also mitochondrial genome are covered at high depths, allowing us to detect a low level of a heteroplasmic variant. And in addition, whole genome sequencing can detect all type of almost variants such as SMV, indel, CMV, structure variant, repeat expansion, while WES can detect only SMV, indel, large CMV. And for structure variant, whole genome sequencing can identify exact breakpoint of almost structure variant, but WES can estimate it in approximate interval of potential breakpoint. So therefore, WGS can replace exome and microarray altogether and find the additional variant that two tests cannot identify. Okay, let me walk you through 3B Genome's workflow from the sample collection to the delivery report. So it all starts with a physician or the researchers ordering our genome and collecting the sample. So for whole genome sequencing, we currently accept whole blood and dry blood spot, and high quality genomic DNA extracted from the specimen. Once a DNA is extracted and quality checked and library preparation is made, sequencing is performed on Illumina Nova 6000, and we run 150 base pair paired end sequencing. So mean depth coverage is 30x with more than 95% of the entire autosome are covered at 20x, and mitochondrial genome is covered at over 1000x. Once sequencing is completed, read are aligned to the human reference genome 38, and variant are called. We adopted GATK for small variant calling, and for structure variant calling, multiple programs are used to detect the CMVs, inversion, translocation, mobile element, and repeat expansion. And once the variants are called, they are annotated, filtered, classified, and prioritized by their pathogenicity and symptom similarity using our own developed system evidence, as I described earlier. And our variant interpretation team reviewed the final list of the rare variant with assistance of 3S, as I described earlier, to determine which variant to report as a potentially disease-causing. At this step, we use another homebrew system, Jebra, but I cannot show how Jebra works. But if you want to see how Jebra works, so you can come to our booth, so I'll demonstrate it. Okay, so we generate a report. Here's an example of the report. Actually, we have 10 sections in the report. The main section is the result section and interpretation section. Result section is summary of the variant and disease association. So result is reported as positive, inconclusive, and negative according to the level of variant pathogenicity and clinical relevance to the patient. And interpretation section provide further details of a reported variant in the context of the HCMG guideline. So let me show the few interesting cases that we solved with WGS as a part of a large and government-funded one-year pilot study that our company participated in 10,000 genomes from more than 4,700 rare disease families just 1 year. This is a summary of these projects. So many patients had exome or the microarray prior to enrolling this project for whole genome sequencing. The overall diagnosis rate approximated 32%, with additional 5.2% of the patient receiving the inconclusive with VS and or 1P or LP variant in a recessive disease. So as you can see the right graph, the disease diagnosis rate can vary quite a bit among disease category. So excluding the adult cancer patient with the lowest diagnosis yield, so diagnosis rate went up to 40%. So of the diagnosed patient, 26% of the patient received a structure variant, and also notably, 4.8% of the patient could only be diagnosed by exome, could only be diagnosed by genome sequencing. So a variant reported include small CMV, not identifiable or not detectable by array or the whole exome sequencing, and repeat expansion identified in the non-coding region, and complex structure variants such as copy neutral, inversion, or translocation, and mitochondrial variant with low level of heteroplasmic, and the deep intronic variant shown or predicted to alter splicing. Okay, let me show the 4 cases. First case is a 60-year-old male with hearing loss. So actually, he had initially exome sequencing. So we found that one single heterozygous frame shift variant in OTOA gene. So we report it as inconclusive because this heterozygous variant was found in a recessive disease. So physician requested the protoband-only whole genome sequencing to look for a second variant. So unfortunately, we are able to find large inversion, which is clued by blue and cyan colored read, suggesting unexpected direction. So based on the lead, so we are able to figure out that there is a heterozygous inversion starting from intron 17 of OTOA gene. So he was diagnosed with hearing loss caused by potential compound heterozygous SMV and inversion of OTOA genes. But parent testing need to confirm if these variants are in trans. I think most likely they are. Second case is 6-year-old male with multiple cafe area spots, suspected of having neurofibromatosis type 1. But initial exome was negative, and there is no clinically significant coding variant in the coding region of NF1 and NF2 genes. The physician requested 3-year whole genome sequencing. So potentially significant non-coding variant was identified in between exon 14 and exon 15 in the NF1 gene. So as you expected, this variant position was not covered by exome. So as you can see that graph, the upper panel is the WGS coverage and lower panel is WES coverage, and the blue highlighted in the blue region is variant position. So there is a many read aligning across the variant position in the WGS, but while WES covers, there is no read. So these variants actually confirmed to be de novo by trio genome sequencing. And also, this variant have been reported in other patients with neurofibromatosis previously. So he was diagnosed with NF1 type 1. And third case is the 2-year-old male with global developmental delay and microcephaly. So similar to the first case, this patient in the exome also, we found the one pathogenic frame shift variant in VPS13B gene. So we report it as inconclusive because this only one single heterozygous variant was found in a recessive disease. So physician request to trio genome sequencing to look for a second variant. So we are able to find the single exonic heterozygous deletion in the same gene. So as you can see that this upper panel is WGS coverage, and the lower panel is the coverage of WES data. So you can see the WGS coverage is constantly uniform across the entire intronic lesion, flanking exon, deleted exon 30, but while WES only the coverage of exon 30 is covered. And also you can see that WGS coverage is constantly half across the yellow highlighted region compared to the flanking lesion, suggesting that this is a heterozygous single exon deletion. So we also can identify the exact breakpoint by using soft clip and the split lead information. So we went back our exome data to confirm that exon coverage of exon 30 was lower than other patients, but it was not significant enough to call it as a deletion. So these variants are confirmed to be in trans, SMV inherited from the mother, and single exon deletion inherited from the father. So finally, he was able to diagnose with Cohen syndrome. Okay, last case. So last case is 1-year-old male with cryptocalcism and muscle weakness, and family history is non-specific. Initial exome was negative. So as you can see that, so in the WGS data, you can see a little peak in there is suggest of the increase of mean depth of coverage. So this region is well described the UTR repeated region in 3' UTR of the MPK genes. But in the exome, there is no read actually because our exome kit does not cover the 3' UTR. And also we're using other tools to predict repeat number. So these tools identify the heterozygous repeat expansion repeated to 123 repeat expansion. And also which must have been inherited from the mother, predicted to carry 90 CTG repeat, while the father had a normal repeat number. So actually he was diagnosed with congenital myotonic dystrophy. Okay, so these 4 cases I show illustrate well how whole genome sequencing can be essential in undiagnosed patient from the exome. So actually this is a common workflow for diagnosing the rare neurodevelopmental disease, a very traditional workflow. So first, microarray is firstly performed to detect the larger CMV, but the CMA's diagnosis rate approximately 10% or 15%. But if it comes back negative, usually larger gene panel or the exome sequencing is the next option. So diagnosis rate is reported to be 25%, but up to 50% depending on the disease category. And then if comes back negative, so it is common practice to wait 1 or 2 years and get exome reanalyzed because the diagnosis rate from the reanalysis can be increased to 10%, usually most from the novel gene discovery. And if the reanalysis is negative, so it is whole genome sequencing is pretty reasonable steps if available, and the diagnosis rate can be increased at 10% by whole genome sequencing. It is mostly from the largest structure variant. So whole genome sequencing is a comprehensive genetic testing with the highest yield. But why are we going through all the other tests before getting to the whole genome sequencing? So usually one actual reason is only cost, I think, yeah. So even though the cost of the sequencing has dramatically reduced in the past 15 years, but the cost of the whole genome sequencing is still $1,000 per sample for just sampling. But however, the cost of the whole genome sequencing is expected to drop even further because you probably heard of the Illumina announcement of new sequencer NovaSeq X, which allowed us to sequence the entire genome for about $200. But of course, the whole genome sequencing, the analysis cost is much higher for exome, but it means that it won't be that long before whole genome sequencing can replace completely whole exome sequencing, I think. Okay, so this is a summary of my talk. So this I'm proposing as genetic testing becoming more and more affordable and readily available. So physicians examine the patient and take the history and learn the basic study. And if the patient is suspected having of genetic disease, instead of the more routine test to figure out the diagnosis, just to perform the genetic testing, preferable by exome or genome rather than single gene or panel testing. And then, guided by the result, the physician performed the additional phenotyping, and then family screening or targeted treatment can be done as indicated. I think this message is what other previous speakers are trying to give out, and I hope this audience will also agree with this message. Okay, this is my last slide. So I'd like to thank my colleagues of 3 billion. So this is a real photo of the people working to create library and load the sequencer, create the variant and customize report and communicate with the doctors and physician researchers and developed algorithm or web interface and server. And so all to diagnose someone with rare genetic disease until the end of the diagnosis odyssey. Okay, thank you for your attention. Do you have any question about the last session?
expect that if you roughly sequence 30 reads or above, you might have trouble to pick them up, either if they are single nucleotide variants or even CMV.
Yeah. Yeah, actually, we use GATK best practice for small variant calling. This is optimized for germline variant. So we have a limitation to detect the somatic mosaicism, and also we do not validation study to confirm the level of limit of detection of the mosaic variant. So, but in my experience, we can detect the 10% of the variant allele friction, the variant. But there is a chance that we missed some variant. But if you are if you have a patient with suspected having a mosaic variant, so if you give me some genes, we can call these variant in genes using other caller, somatic caller. But there's also limitation. You know, the whole genome sequence covers only 30x. So I think the very extremely low level of the allele frequency, the variant, we can miss that. Yeah.
Can you accept DNA biopsies for suspected somatic mosaic disorders?
Sorry.
If we did a skin biopsy or a vascular biopsy, can you extract?
Oh no, not yet. Okay.
If I may, a second question. You said that you got impressive 8% of case solutions by structural variants. What's your experience picking up structural variants using short-read genomes?
Yeah, very good question. And when we review the structure variant, it is very very labor-intensive process. You know, by using prioritization tool, we have reviewed all the variant to find the exact breakpoint. But actually, we have internally 10,000 genome database. So actually, we can filter the common variant, and then we classify these variant according to the ACMG guideline. So, and then maybe 10 or 15 variant remained. So we can compare the patient phenotype and between the patient phenotype and the symptoms associated with the disease. Yeah. Yeah, any other question? Okay, thank you very much. And lastly, I want to announce the special promotion for the ESHG. So this exclusive opportunity give you to receive Exome and Genome for a discounted price of $990. So in the event of NGS negative Exome result, the complimentary WGS is performed immediately and no additional cost. So please scan the QR and register our website, and then you can order that. And okay? Yeah, and also we have a special gift for you. So if you get the blue mark in our booth and the red mark at our symposium, so it is eligible for the special gift. It is Korean traditional thing for summer. Okay. And we sincerely appreciate your presence in the early hour. And I hope all of you have a pretty good understanding of what 3 billion does. And this knowledge gained from these 3 sessions will be greatly benefit from your clinical practice. And if you are not already, so I hope all of you become our partner in exploring rare genetic disease. Thank you for your time.